This lecture will cover how compounds are identified as potential drug candidates. It involves the identification of hit compounds, which are identified by showing a positive result in an assay. Not all of these molecules will have an effect at the target, so they are investigated in further assays to narrow down the compounds to leads. Leads are compounds that have the desired effect at the target and have properties that make them suitable for development into a drug. In the lecture, you will learn about a number of topics. It is important to know what is meant by the terms active, hit, and lead, and how these terms are related, in particular, at what point a hit becomes a lead. You should also understand what properties a compound should have in order to be considered a good lead and be suitable for development into a drug. There are several different sources of compounds that can be investigated in the search for a lead. You should be able to discuss these sources and have an appreciation of the numbers of compounds that each type of source can provide. A lot of compounds may already be available, for example, natural products, and compounds that have been produced from previous drug discovery projects. Completely new compounds can be generated by a process known as combinatorial chemistry, which you should be able to explain. High throughput screening is at the core of all industrial drug discovery programs, so it is important that you understand the concept and can explain, in an exam, how it is used in generating new lead compounds. You should also gain an appreciation of how a screen is designed and validated. So now I'll give you the definitions for actives, hits, and leads. A biological screen employs an assay designed to detect an interaction between a compound and the drug target. It could be, for example, a binding assay. At this early stage, very large numbers of compounds will be screened, so the assay needs to be able to assess compounds rapidly. A positive result at this stage does not mean that the compound interacts with the target in the desired way. For example, the compound might bind to the target, but it causes denaturation of the target rather than acting by a pharmacological mechanism. It may be that the drug discovery project is searching for a receptor antagonist, but the compound showing a positive result in the assay is an agonist. The compounds showing a positive result at this stage are therefore defined as actives. Once actives have been identified, they are further screened using an assay that can distinguish how the compound affects the target. This may be, for example, a functional assay designed to identify compounds that bind to the receptor and act as an antagonist. Once a compound has been shown to interact with the target by the desired mechanism, it becomes a hit. But just because a compound is a hit, it does not mean that it will make a drug. For example, it could be toxic, or it may not be absorbed well by the body, or it may be metabolized by the body to such an extent that it cannot reach effective concentrations in the blood. A lot more is needed to be understood before a compound can be considered as a drug candidate. Lead compounds are hit compounds that have structures that can be optimized by chemists to produce a candidate drug. Compounds may be acquired by synthesis or from commercial or other sources. Enormous numbers of compounds are involved, and it is vital that no mistakes are made when compounds are identified as hits. Therefore, there needs to be foolproof methods of storing the compounds and controlling the stock. For a compound to bind to a target, it needs to be in solution when delivered to the assay. So, as compounds do not all dissolve in the same solvents, their solubility needs to be determined. A screening cascade employs a series of assays to identify actives, then hits. Mistakes at this stage can be misleading, so time is invested to ensure that the screen is of high quality. That means that the assays will only detect compounds that interact with the target, and actives and hits will be detected reproducibly. Once the compounds and screening cascade have been validated, primary screening can begin. The first assay in the screen identifies actives, which are taken into further assays to identify hits. The properties of the hits are then assessed to narrow down the compounds to leads. These early screens may be qualitative in nature. In other words, they are designed to check if the compound has an effect or not, at a concentration that would be deemed appropriate for a pharmacological action. The next step is to investigate the leads quantitatively for efficacy. This involves testing them over a wide range of concentrations. Once a lead has been identified, it is time to seek a patent. First, a search must be done to ensure the compound has not already been patented. Then a patent is applied for to protect the intellectual property or IP involved in the discovery of the compound. Many compounds protected at this stage may not make it all the way to becoming a drug. But it is important to protect the IP as early as possible to stop competitors getting there first. The process of generating a lead depends critically on medicinal chemistry, but it generates a lot of structures and data that must be managed, so it's also heavily dependent on informatics. So how does a hit become a lead? There are several criteria that must be fulfilled for a compound to reach this status. Obviously, it should have the desired pharmacological properties and produce the right effect in the screens. But it does not need to be particularly potent. Once a basic molecular structure has been identified, it can be tweaked by chemists and structure-activity relationships studied in order to improve its potency and/or efficacy. It is important that the compound is not extremely polar or hydrophobic, because this can prevent uptake of the drug into the body, and small tweaks to the structure may not be able to rectify it. We know quite a lot now about the types of chemical groups that can cause toxicity, so a lead compound should not contain any of these toxic groups or groups known to produce toxic metabolites when administered to the body. A property that is desirable, but not necessarily essential, is that the compound should interact reversibly with the target. In most cases, reversibility is important, but an irreversible interaction can sometimes be advantageous. For example, the irreversibility of block produced by aspirin at the cyclooxygenase enzyme is the reason that it is effective at preventing platelet aggregation, making it useful at preventing blood clots. Some penicillins target bacteria, and the irreversible nature of their action is clearly beneficial. A range of different sources of compounds are used in screening for new drugs. One of the major resources is compound libraries. These are stores of compounds held, in most cases, by drug companies. They are kept in much the same way as a library stores books. These photographs show you what a typical compound library looks like. Compounds are stored in bottles on shelves. They are carefully catalogued with barcodes, which are read by robots that automatically collect the compounds to be tested in a screening cascade. In recent years, companies have begun to make their compound libraries more widely accessible by sharing them for a price. There are also companies that specialise in maintaining compound libraries and selling access to them. Compounds are also available from academic sources, often generated as part of specific research projects. These tend to be much smaller than commercial or company libraries. Compounds can also be synthesized from scratch. This may be on a small scale, for example, in an academic laboratory, but it often involves synthesis on a grand scale, using a process known as combinatorial chemistry, which we shall explore shortly. Another source of new compounds is natural products, or chemicals produced by living organisms. Some of the earliest drugs, such as aspirin and digoxin, were isolated from plants, but but there are many additional sources of natural products. The newest source of compounds is biological agents, such as antibodies and proteins from recombinant DNA technology, as we covered in an earlier lecture. You've already been made aware of natural sources as drugs. There's a long history throughout the world of the use of plants to treat illness. For example, morphine from the opium poppy is still our most effective drug to treat pain. The ability of plant constituents to modify behaviour is also widely recognised. For example, opium and cannabis from hemp. As well as plants, drugs have been obtained from bacteria, fungi, or moulds, for example penicillin, and animals. Venomous animals are a particularly rich source of potential leads. Examples include toxins from snake, spider, scorpion, and amphibian venom, as well as a range of marine animals. The constituents of venom, usually proteins involved in generating the poisonous effect, often have very specific interactions with drug targets. Drugs may be isolated directly from living organisms, or molecules from these sources can act as a lead compound or template for synthesizing new compounds. Natural products have, however, been widely studied over centuries, so although new pharmacologically active molecules continue to be discovered, the rate of discovery has slowed. As more are found, there are fewer left to be discovered. I told you earlier about combinatorial chemistry as a method to synthesize new compounds for screening. This is an approach to synthesizing a large number, tens of thousands to millions of compounds. The compounds generated are all structurally related due to the way in which they are generated. The basic principle of combinatorial chemistry is illustrated here. It starts with two series of simple structures, which are reacted together to form more complex structures. We shall denote the starting structures as Series A and Series B. In this example, Series A is based on a benzene ring with a carboxylic acid group attached. There are three members of the series here, each with a different number of methylene groups separating the benzene ring from the carboxylate group. The black triangle represents the structure with no methylene groups. The green triangle is with one methylene group, and the red triangle with two methylene groups. The Series B structures are simple organic molecules with a variable group R1, R2, or R3 attached to the central carbon. These are represented as black, green, and red dots. When these two series of molecules are mixed together, they react to form a mixture of compounds, with the general composition illustrated here, for the product of the reaction between the black triangle and green dot. When the Series A and B molecules are mixed together, each of the A series molecules can interact with any of the B series molecules. The reactions therefore produce a mixture of 9 members of a new series, made up of each possible combination of the starting points. This is known as a 3x3 library. This is a relatively simple example, but more complex mixtures can be made by adding a third series of structures, or including more members of each series. So, for example, a 20x20x20 library would give a mixture of 8,000 structurally related compounds. This approach can also be taken to prepare libraries of proteins. Hexapeptides, or peptides containing 6 amino acids, are often used. If all the possible hexapeptides from a set of 20 amino acids are added together, the number of proteins contained in the product will be 64 million, or 20 to the power of 6 possible combinations. It would be impossible to synthesize each of these individually. The mixtures of compounds are applied together in a screen. If there is no hit, the sample can be discarded and multiple compounds are ruled out. But if there is a hit, samples can be synthesized and tested in cycles containing samples with increasingly less complexity. Once you have identified a sample containing a hit, you need to redesign the synthesis to work out what molecule in the mixture was responsible for the hit. This slide illustrates one of the approaches to doing this, called mix and split synthesis. Instead of mixing all the series A and series B molecules together, all the A components are mixed. The resulting solution is split in 3, and each of the series B molecules is added individually to each of the solutions containing the mix of A molecules. The result is 3 test solutions, which each contain only 3 combinations of A and B precursors. Testing these mixtures in the assay will further narrow down the cause of the hit. The 3 products in the sample could then be synthesized individually and tested again to finally identify the hit. Multiple parallel synthesis is a method with which solutions containing individual combinations of series A and B molecules can be made. Because of the potential large scale, involving possibly thousands to millions of compounds, this approach is critically dependent on having automated methods, that is, robots, for picking up and mixing solutions, processing the product, and analysing the data. This synthesis is usually carried out in a rectangular plate containing a matrix of wells. The process begins with the starting molecules from the A series, which are distributed in the wells such that each row contains a separate A molecule. Each B series molecule is then added to a separate column of wells. In this way, each well contains a unique combination of different A and B molecules. Considering all sources of potential drug molecules, pharmaceutical companies generally have access to hundreds of thousands, maybe several million compounds for screening. The number of test compounds from each source that is available for screening varies widely. The largest number is found in historic collections of compounds or compound libraries. As they have usually been accumulated over long periods of drug discovery activity, they can contain upwards of 100,000 compounds. As you have seen, combinatorial chemistry can produce tens of thousands to millions of compounds, and as they are made, they are added to compound libraries. Natural products are a potential source of much fewer compounds, estimated to be no more than a few tens of thousands. Despite the large number of compounds available for screening, new therapeutic agents appear at a low and reducing rate. This relative lack of success in developing new drugs from small organic molecules is partly why pharmaceutical companies have become more involved in developing biologic agents using new molecular tools. While it is important to have large numbers of test compounds available, the diversity in the structures of these compounds is probably more important. That is because with diverse structures, the chances of having a molecule that recognizes a new target is higher. So let's look at how these different sources of chemicals compare in terms of diversity. One of the problems that companies have is that their libraries can lack structural diversity. Libraries very much reflect the company's history. So, if it spent many years developing, say, beta blockers to treat cardiovascular disease, the library may contain many compounds with activity at these receptors, but they would inevitably not be hugely different. Drug discovery programs were running when we knew a lot less than now about what gives a compound drug-like properties. Consequently, historic libraries may be limited in molecules with properties that make them compatible with becoming drug candidates. The recent trend towards sharing of libraries may help with these restrictions. By design, combinatorial chemistry produces related structures based on simple starting molecules. Diversity can be introduced into these structures by clever design of the starting molecules and varying the starting molecules in different rounds of synthesis. The quality of the compounds very much reflects good design and the quality of the chemists. Structural diversity is where natural products excel. Nature can produce some remarkable compounds with unexpected and unpredictable structures. An example is a protein-based glue secreted by a frog onto its skin where it traps biting insects, which it can then eat. The glue is stronger than available medical glues, and it's being investigated for medical and other uses. A problem with natural products is that they may not be consistently produced in the same species, for example, in different seasons or environmental conditions. Depending on the rarity or origin of the species, it may also be difficult to get a reproducible supply of the plant or organism. Because the chemical depends on the supply of the organism, it may also be difficult to scale up the production of the compound to industrial quantities. Once the structure of molecules identified from natural products is known, then it may be possible to generate them industrially using synthetic chemistry or biological technology. Compounds from different sources provide the starting point for screening, with the aim of generating a lead compound. How you go about screening will depend on what information you have about the drug target and the availability of compounds already known to interact with the target. There are two basic approaches: rational design and empirical screening. Rational design depends on there being either knowledge of the target structure, an endogenous ligand for the target, or a synthetic compound that interacts with the target. If ligands are already available, then analogues of these ligands can be designed, synthesized, and screened in a target-based assay. An example of this approach was the discovery of methanacol, a synthetic analogue of acetylcholine, which is used to treat bladder dysfunction. It's a muscarinic agonist that is resistant to metabolism by acetylcholinesterase. Often, the first drug to be discovered in its class can be improved on. For example, the first calcium antagonist to be discovered and used to treat hypertension was nifedipine, but it had to be taken twice a day to maintain its effect. Using nifedipine as a template to generate more analogues for screening, new calcium antagonists were discovered with longer duration of action. Drugs like amlodipine, which can be taken once daily, have now superseded nifedipine in the clinic. This is an example of the fast follower approach. In silico studies of the target structure may be able to suggest structural components likely to interact with the target. Molecules for screening would be designed around the suggested structures. If there is no information available about the structure of the target and no ligand structures to direct the design of molecules for screening, the only option is empirical screening. This involves screening any libraries of synthetic or natural compounds that can be obtained. This approach makes no assumptions about what type of structure might be active. The process of rational design begins with information from computational chemistry and structural biology. This information is used for molecular modelling done in silico to suggest structures likely to interact with the target. If there are known ligands, then ideas about potential structures will already exist. A range of compounds are synthesized based on the modelling or ligand knowledge. They are then screened for activity in a cascade of assays which are designed to detect the desired activity. The results are then reviewed. At that point, a compound may be identified as a lead and taken forward to the lead optimization stage. Alternatively, based on new information on the types of structures that produce hits or not in the assays, compound structures may be further modified and screening repeated until a lead is generated. With the amount of structural information we have about targets increasing all the time, rational design is becoming an increasingly viable option. It is also an attractive approach when drugs that interact with the target are already available. Drugs are usually low molecular weight molecules that can be used as a starting point for designing and synthesizing new analogues. Rational design is also an option when there is mechanistic information available about binding sites on the target and how molecules can influence target activity. In this case, the molecules might be large proteins or toxins as opposed to drug-like small molecules. Such large molecules might not be suitable as drugs, but by studying their interactions with the target, binding sites for drugs, and therefore potential drug structures, could be predicted. Target class analogies are exemplified by the use of nifedipine as a template to search for better calcium antagonist drugs to treat cardiovascular disease. The aim here is to identify a best-in-class drug. The alternative approach is to search for a first-in-class drug. In other words, new drugs can be molecules that act by the same mechanism as existing drugs but are better. Or they could be the first example of a class of drugs that work by an entirely new mechanism. At the start of empirical screening, there may be no information available about binding sites on the target, and no chemicals available that interact with the target. So there are no ideas about what kinds of structures might bind to the target and have an effect. If this is the case, then every compound available for screening will be tested in high-throughput screening, with the aim of finding initial hits, which will direct future screening. Or, if there is some limited structure activity information available, screening will begin with molecular structures that are consistent with the knowledge available. This may still mean screening thousands of compounds to identify initial hits, but there may be enough information available to rule out certain types of structure, even if there is not enough available to narrow the design to particular analogues. The structures of the hits identified from the initial screen will direct the synthesis of new compounds. At this point, the screening strategy takes a more rational approach. Compounds are screened by testing for activity in a cascade of assays designed to detect target activity and assess chemical properties. The results are then reviewed, followed by further synthesis and testing until a lead is identified. It will then be taken forward to the lead optimization stage. The main benefit of empirical screening is that it can identify low molecular weight compounds that can be developed into drugs even when nothing is known about the structure of the target or what might interact with it. Success very much depends on having access to good compound libraries which, if you are lucky, contain active compounds. The empirical approach can be used even when structural information about the target is available. In this case, the advantage is that unpredicted structures can produce hits. This can start a new area of research, leading to information on unpredicted and completely unexpected structure-activity relationships. It is also an approach to exploiting unique collections of compounds which increase the chances of a patentable discovery. Because the automated screening of large numbers of compounds is relatively quick and efficient, this approach can give a company a competitive edge. The screening of compounds in the pharmaceutical industry employs high-throughput approaches. large number of compounds are tested in a coordinated series of assays designed to identify actives, hits, and then leads. It is a highly automated process using robots to carry out almost every step once the assays have been validated and the compounds for screening prepared. The process is known as high throughput screening. The key requirements for a high throughput screen are listed here. There are videos on high throughput screening on Blackboard. You can find the links in the course information page and in the lecture folder for this week. Viewing the videos should give you a better feel for the technology involved and how screening cascades are organised. High throughput screening uses plates containing a varying number of wells. The plates are 12.8 centimetres long by 8.5 centimetres wide by 1.4 centimetres deep. The capacity of assays in high throughput screens has been steadily increasing. Around the mid-1990s, the available plates contained 96 wells, each with a volume of 350 microlitres. For any target, this allowed around 10,000 compounds to be tested each week. From the late 1990s until now, plates have contained 384 wells, each with a volume of around 100 microlitres. This allowed the number of compounds to be tested per target to increase to tens of thousands per day. Plates are now available that contain 1,536 wells, each with a volume of 12 microlitres. This will increase capacity to hundreds of thousands of compounds per target per day. This photo compares a 96 well plate at the top to a 384 well plate in the middle and a plate containing 1,536 wells at the bottom. In designing a screening cascade, you need to consider first what the screening strategy will be, for example, following a rational or empirical design. The most appropriate assays to use will be determined by the nature of the target. Before using in a high throughput mode, assays must be set up and tested on a small scale in order to demonstrate the appropriate biological activity. The assay must be validated by establishing proof that the assay measures what is required and is reproducible and reliable. It is then scaled up and optimized for automated use in a high throughput system. It then has to be validated in its final high throughput form to ensure that it is fit for purpose. For a screen to be validated, several criteria must be met. The signal that detects biological activity must be reproducible between wells within a plate and between plates. It must also be reproducible when the assay is repeated on different days with, for example, different batches of cells and compound. Reproducibility must be maintained when the assay is run at the level of throughput that the screening will use, for example, 96 well plates versus 384 well plates. Measurements are made when the screen is run at different times and the correlation between the data from each run established. The long-term reliability of the screen is determined in a similar way, repeating the testing at intervals over a prolonged period. It is also essential to establish that the screen is robust. This is indicated by both the variation in the measurements between assay runs and the dynamic range of the signal measured. The dynamic range is measured as the ratio of the signal to the variation in the background signal or noise above which the signal is measured. This is a summary of the processes involved in lead generation. Starting at the top, compounds are screened and activity detected. The active compounds are screened again to confirm activity at the target and to measure the concentration range over which they are active. This information is used when testing activities in a cell-based assay to check that they have the desired biological effect and are therefore hits. Detection of cellular activity can use a phenotypic assay where the cellular response measured makes few assumptions about the molecular details of how the system works. This is a bit like empirical screening and can identify compounds that affect complex cellular systems that are poorly understood, so clearly established targets are not available. Examples might include CNS disease, such as depression, where it is not yet clear what the best molecular targets might be. Where a clear target has been identified, the assay would employ a mode of action endpoint, such that the signal indicates, for example, whether the compound acts as an agonist or antagonist. Having identified hits, their physicochemical properties would be investigated to determine if they fulfill the criteria for lead compounds. Leads would then be investigated in further assays for a variety of properties, including kinetics and selectivity of action. As the discovery process progresses from the compound library through to generating leads, there is enormous attrition in the number of compounds involved. Most of the compounds tested have no effect on the targets they are screened against.